Hello and welcome to this next Hello and welcome to this next video on OCR A-Level Biology. This is module 3.2, which is biochemistry. Now, before we get started, I want to say that this is a long topic. So what I'm going to do is split this into 3 videos. In this first video, I'm going to focus on water and carbohydrates. In the next video, I'm going to look at lipids. And in the final video, I'm going to look at proteins and inorganic ions. So, we'll start with just a basic introduction to biochemistry. So there are 4 major biomolecules that we need to be aware of. Well, 5 really. But DNA and RNA are in a separate module. So, the first one is obviously water. The second one is carbohydrates. The third one is lipids. This next one is proteins. And we also have DNA, RNA, so nucleic acids. But we won't worry about them for this topic. So in this first video, I'm going to be only focusing on water and carbohydrates. So first up, let's talk about water. Now water, well, if you do A-level chemistry, if you do chemistry at A-level, then you will know that water is a polar molecule. So water is this molecule here. So, and actually water is considered a polar molecule for a few reasons. This oxygen you can see here, it is slightly electronegative because it has more protons than these hydrogens. So those protons would want to attract these electrons that are within the covalent bond here. So what does this do? Because it has a greater number of protons, it exerts a stronger attraction on the said electrons in this covalent bond. That means that this oxygen becomes what we call delta minus. Delta meaning a very small change. As a result of that, this hydrogen here, that you can see, whoops, where's my pen, becomes delta plus, because it's losing these said electrons to the oxygen. So, if we put this water molecule into solution with a solute, let's say with NaCl, what will happen is these hydrogens, because they're delta plus, they could be slightly attracted to the Cl minus ion down here. And this water, this oxygen, because it's delta minus, it's going to be slightly attracted to the Na plus over here. These are what we call polar bonds. And that's one of the properties, and this is what makes up one of the properties of water. And one of those properties is a solvent. It's a good solvent. Because water is able to form these polar bonds with the solute, with ions in the solute or the salt, it makes it a very good solvent. But water is not just a very good solvent. As you can see here, water can form polar bonds with itself. So here we have something, we have 1, 2, 3 water molecules here, and these dashed lines that are occurring between this electronegative oxygen or delta minus oxygen and this delta plus hydrogen, this is another special type of polar bond, and this is called a hydrogen bond. So a hydrogen bond is a weak, it's not too weak like van der Waals forces, but it's slightly stronger than van der Waals forces, but it's is a weak interaction when molecules are where slightly negatively charged, wherever molecules contain, such as water, contain a slightly negatively charged atom, such as oxygen, bonded to a slightly positively charged hydrogen atom. So that's what you can see here. This hydrogen atom is slightly attracted to this delta minus oxygen. And because of that, water has some very intriguing properties. So one of those properties, as we mentioned, is a good solvent. Another property that we have here, sorry, I need water, 1 second. Another property is it's a very good reactant. So if I get rid of this for 1 second, if I move this over here, actually I don't need that anymore. It's a very good reactant. So this diagram will help to explain it. Now, there are two types of reactions we need to be aware of. Let me zoom in just on this. So if we zoom into this, we can see why. So here we have two molecules, and we have both of these molecules have OH groups. What happens is when these OH groups are close together, basically these two molecules will join together to form molecule 3. And when that happens, water is released. So water is released. Whoops, let me just move. So water is released out when this hydrogen here and this OH combine to form water, and then it will form a bond between the oxygen left here on molecule 1 and molecule 2 to form molecule 3. This is called a condensation reaction. Condensation is joining together, but it's also losing water in the process. Now if we want to split up molecule 3 into its respective molecule 1 and molecule 2, we need to add water in. So we add water in to break this bond here, to form the two OH groups that were in molecule 1 and molecule 2. So this is called a hydrolysis reaction. Hydro means water, lysis splitting, so it literally means water splitting. Okay, so that's why water is a very good reactant. There are some other properties of water that we need to be aware of. For instance, we have something called cohesion. So water has something called cohesion. So if we go over here, hopefully I have enough space. This is cohesion and surface tension. Basic cohesion means because those water molecules are able to form hydrogen bonds with each other, the hydrogen bonds basically pull the water molecules together, which is why a drop of water, if you spill like a drop of water onto like a surface, that drop of water will not like immediately spread out. It will like stay contained within its drop of water, I guess. So this pulling together of water molecules due to hydrogen bonding is called cohesion. So cohesion is the pulling of water molecules together because of this hydrogen bonding. I'll probably explain that a little bit rubbish, but if you want more information, go and look at page 52 of the textbook. So, this cohesion also happens at the surface of water as well. And when this happens at the surface of water, we call it surface tension. And surface tension is what allows things like these pond skaters to be able to walk on water. And also in plants, I'm not a plant expert here, but it also allows columns of water in plant vascular tissue to be pulled up by xylem together as one big column from the roots to the leaves in transpiration, in order to get that water to all parts of the plant in transpiration. So that's another property. Another property is the density. Of course, density. You may have already experienced this. Density. Ice is less dense. Ice floating on water. You may have experienced this before. So, the density provides an ideal habitat for specifically aquatic organisms. So, you probably all know that ice, when water is frozen in ice, it floats on top of water. So solid water floats on top of liquid water. That's because ice is less dense than water. Because of that, the polar structure, because of its polar nature, these water molecules, when you freeze water, it's less able to spread them out apart with lots of space in between in a very rigid structure, but with lots of space in between the molecules due to these hydrogen bonds. So that's because ice is less dense than water. And that is why ice is less dense than water. And this is critical for aquatic organisms because they have a stable environment, it provides a stable environment. And because ice floats on water, if the top of a lake or a pond or a river is frozen solid, that ice can actually help insulate the water below against extreme cold, so that it helps aquatic organisms below the water to survive. So if you didn't get what I just said right now, basically because if you have ice floating on top of if you have like a frozen lake, while the ice is on top of the lake, like frozen ice on top of a lake or a pond or a river, maybe even the sea, that ice helps insulate the water below and helps provide a nice environment for aquatic organisms to thrive in or to survive in. Probably if you didn't get that, then again look at page 52 of your textbook. And then some other things that I'm not going to go into much is here we have two other properties here. Here we have specific heat capacity, so it's a high specific heat capacity. And here I'm not going to go into these in much detail. Basically it allows aquatic organisms again have a stable environment in which to live. If you want to know what it is, it's basically the amount of heat energy required to raise the temperature of 1 kilogram by water by 1 degree Celsius. And the specific heat capacity of water is 4.2 kilojoules of energy required per kilogram per degree C. And then we also have a high latent heat or vaporization. And because of that, because of the hydrogen, how strong the hydrogen bonds are, water has a high boiling point. So it has a high latent heat of vaporization. So a large amount of energy is required to create those hydrogen bonds to turn water into steam. So therefore water can help us, as humans, help to cool mammals down. For example, us, when sweat evaporates, it takes away some of that heat energy. And for plants, when water evaporates from mesophyll cells, it helps the plant to cool down. And that's basically all you need to know about water. If you need any more information than what I just said, please look at page 52 and 53. There's a pages 52 and 53 give a detailed description or detailed analysis of the properties of water, I guess. Okay, so that's water. So that's the first molecule that we're looking at in this short video. Remember, I did say this topic is quite long, so I'm going to split this into 3 videos. So in this video, we've already done half of this video, which is water. We're now going to do the other molecule we're looking at, which is carbohydrates. So now we're going to look at carbohydrates. So carbohydrates. There are many different types of carbohydrates. So carbohydrates are basically sugars, and they have a variety of functions. They are the main source of energy. They're the main source of energy in aerobic cellular respiration, for example glucose, and they can be used as a store of energy as well. So, and sometimes they're found in nucleic acids. They're used to provide structure support. They have many different functions, but the main function that they have is they're the main source of energy, and sometimes as a storage of energy as well. So there are different classes of carbohydrates. So we have monosaccharides. Disaccharides. This one you don't really need to know, but oligosaccharides and polysaccharides. So monosaccharides are the first one that we're going to talk about. Monosaccharides. So monosaccharides, these are the simplest carbohydrates, simple sugars. They're referred to as simple. Whoops, I do not need to write in blue for this one. So monosaccharides are classed as simple sugars. And they're simple sugars. They're basically the building blocks of all the other types of carbohydrates. Building blocks. Or monomers. The more fancier term is monomers of carbohydrates. And there are different types of monosaccharides that you need to be aware of. So there's 3 main classes, well 4 main classes really. We have triose sugars, which is a carbohydrate with 3 carbons. We have tetrose, which is a carbohydrate with 4 sugars. We have pentose, which is a carbohydrate with 5 sugars. And we have heterose sugars, which is a carbohydrate with 6 sugars. So triose means 3, tetrose is 4, pent means 5, so 5 carbons, heterose, means 6, so 6 carbons. So we're mainly focusing on heterosugars in this topic, and particularly in this video. We'll focus on the pentasugars when we do DNA and RNA. So the heterosugars, there are four different types of heterosugars that you need to be aware of. So this first one is galactose. So galactose is found in lactose, as you may know. Is found in milk. We have fructose. And we have two types of glucose. So we have alpha glucose. And explain why it has this fancier alpha term in. And we have beta glucose. Now, going back to alpha and beta glucose. And going to alpha and beta glucose. Basically, an alpha glucose molecule is where the OH of this, I can to show you here, where this OH is facing in the opposite direction to the CH2OH, which is up here. So that up here, so the OH of carbon 1, so when we look at glucose, we label the carbons like this, if I can draw it. So we go clockwise from the oxygen, CH2OH. So when we put numbers on, we number the carbons from the oxygen doing clockwise. So this would be carbon 1, this would be carbon 2, 3, 4, 5, and then this is carbon 6 here. So in alpha glucose, which is what this is, so this is alpha glucose. We have here the OH of carbon 1 is pointing in the opposite direction of the OH to the OH of carbon number 6. So carbon number 1's OH is pointing in the opposite direction to the OH of carbon number 6. So if I draw beta glucose, so it's the same for alpha, so we can also get alpha galactose and alpha fructose as well. For beta glucose, and actually any beta monosaccharide or beta carbohydrate, I'll actually draw it here. It's basically the same structure, but if I draw it out, CH2OH, you can see a slight difference. You can probably already see right now. This is where, so this is beta glucose. So again, if I number these molecules, so 1, 2, 3, 4, 5, 6, you can see here that the OH of carbon number 1 is pointing in the same direction as the OH of carbon number 6. So this here is beta glucose. That's how I like to distinguish between the two. So alpha glucose is opposite direction, beta glucose is same direction. Remember, it's to do with the direction of the OH on carbon 1 and 6. So now we're going to look at glycosidic bonding. So that was all on monosaccharides. Now we're going to look at disaccharides and glycosidic bonding. So disaccharides we'll talk about over here in this far section here. So disaccharides, they are formed from two monosaccharides. Joined together They're formed from two monosaccharides joined together, and basically they're formed in a condensation reaction. So here, whoops, if I do this, and if I do move up here, I can show you how this is. So this is the formation of maltose, if you like. If I just zoom in here, and then do 5, 2, no, 1, okay. So this is the formation of a maltose molecule. So we can see here, if I do, let's do orange, since I haven't used orange yet. If we have two alpha glucose molecules here, and basically what happens is the OH of carbon number 1, so let's say 1, 2, 3, 4, 5, 6 there, and the OH of carbon number 4, 1, 2, 3, you won't be able to see these 4, 5, 6. They lose a hydrogen and they lose two hydrogens and an oxygen to form a water molecule in a condensation reaction. So water is released, H2O released, and when doing so they form this maltose molecule. So I'm going to actually try and draw it, which is going to be a little bit of a pain, but we'll try. I'll try actually. Such a small space to fit it in there. So if I do this, actually if I just do that, and then if I do this, you can see here that we form maltose. So this is a two alpha glucose molecules joining together to form maltose. And the bond, this bond here, this is called a glycosidic bond. Specifically, it's called an alpha 1,4 glycosidic bond. The reason it's got this fancier alpha 1,4 thing is because one, these two molecules are alpha glucose, these are both alpha glucose molecules, and two, it's 1,4 because it occurs between the carbon, the OH of carbon number 1 and the OH of carbon number 4 to produce maltose. And so there are so maltose is just, so that, and if we do remove the water, and if we, so that's what happens if we remove water. If we add water back in, we will be able to break this alpha 1,4 glycosidic bond and reform these two alpha glucose molecules in a hydrolysis reaction. Now besides maltose, we have some other examples that we need to know. So examples of disaccharides include alpha glucose when we make maltose, we just said maltose. Alpha glucose and fructose together makes sucrose, and alpha glucose and beta galactose so makes up lactose, and lactose is found in milk, as most of you may know. And two beta glucose molecules make something called cellobiose. Is that how you say it? I don't know. This thing is probably in plants. And me being a biomed student, we don't talk about plants that much. So yeah. So maltose is whoops. Okay. Alpha glucose plus alpha glucose. Sucrose is alpha glucose plus fructose. Lactose is beta gal plus alpha glu. And cellulobiose is 2 times beta glu. That's probably a better way of writing it. So those are all disaccharides. So now we're going to move on to polysaccharides. Well, there is actually one other type of carbohydrate that you need to know about, but it's not in the A level. But you will learn about it when you come to university, and that is called oligosaccharides. I'll explain a bit about it now. So oligosaccharides and polysaccharides are sort of the same thing, except oligosaccharides are less than 20 monosaccharides long, and polysaccharides are more than 20 monosaccharides long. That's all you need to know about oligosaccharides for now. Polysaccharides, we have some examples. So polysaccharides. So polysaccharides, they are made up of 20, as I said, 20 or more monosaccharides, and they're mainly used as energy saccharides, and their main function is to store energy. They act as energy storage. So if you may know that cellular respiration involves this equation, so glucose plus oxygen goes to CO2 plus H2O plus ATP. What ATP you won't know now, you'll learn about it more next year. And there are 3 main types of polysaccharides that you need to know for the exam. So the first one here, I'll write it in red actually, is amylose. So these first 2 are found in plants. This one is found in animals. So amylose. So amylose is found in plants, as I just said, plants. It coils into something called, and is made up of alpha glucose, and the structure of it is it coils. It basically coils into a spiral / helical shape. Coils into helical spiral shape. Shape here. And also now it's made up of hydrogen bonds, so it has hydrogen bonds which hold it in its spiral shape. Stabilize is a better word. Stabilize spiral shape structure. So it's less soluble. Because of these hydrogen bonds stabilizing the structure, makes amylose less soluble in water. So that's amylose, and it has 1,4 glycosidic bonds, as you can see here. The next one is something called amylopectin. This is also found in plants, but it's a bit more complicated than amylose. Plants. It also coils into spiral shape, as you can see, it's coiling sort of here, coils into spiral shape. But this time we have, it's branched. This one is branched. And it contains 1,6 alpha 1,6 glycosidic bonds. And basically this branching means it's able to store a lot of energy, and it's quite compact. It's quite compact. It's more compact than amylose, which is just one straight chain. But amylopectin helps it be a little bit more compact in plants. Animals, however, have a better solution to this storing of energy and making it even more compact. So this one is glycogen. So glycogen, this is found in animals. However, these chains here, 1,4 change, 1,4 change, are slightly smaller than amylopectin. It does have more branching, so it is branched, but it has more branching. And because it has more branching, that means it makes it, let me write this, where is the button for the pen, makes it more compact, very compact. So those are the three main structural polysaccharides that you need to know, which act as energy stores. There is another polysaccharide that you need to know about, and that is, I've drawn them here, and that is cellulose. So cellulose we'll talk about over here. So cellulose. Cellulose, this is only found in plants, so it's only in plants. It's a polymer of beta glucose, and we've just explained the difference between them. So, and I've got structures here to show you again what that means. So this is alpha glucose over here. You can see here that this OH and this OH is this OH here. This is facing in the opposite direction. And actually I don't want to use brown because I have brown up there. Purple. So here these are facing in the opposite direction. So this is opposite for alpha, and this is same for beta glucose. So this structure, if you didn't hear that, this is alpha glucose, and this here is beta glucose. Okay, so beta glucose can also form these glycosidic bonds, but because of its structure, it's unable to form these glycosidic bonds in the same way as alpha glucose. So if we took another beta glucose molecule down here, whoops, that is not meant to be in there, that is meant to be a hydrogen. So basically, when you need So it does still form glycosidic bonds with each other, but you need to invert the adjacent beta glucose molecule in order to form these glycosidic bonds. Because OH and then there's an OH down here, these can't form hydrogen bonds. You can't form a diagonal glycosidic bond. So they need to be like fully like straight on. So what we need to do is we need to invert this next molecule here. So this, oh man, this is going to be really close to draw. So we have an OH here and an H here. So here we have the O. Oh, this is really awkward to draw. So I basically need to draw this kind of shape. So you can see here it's inverted slightly. And let me just do this. There we go. So this is another beta glucose molecule. And when these two join together, you're going to lose. So when these join together, you're going to lose this OH here and this hydrogen here, and then you end up with a bond between this oxygen, whoops, so these two will come together to form water. And what happens is you end up with a bond between this oxygen here. Oh my god. You end up with a bond between this oxygen here joining to there. And I'll show you what that looks like in practice. Whoa. Yeah, you basically end up with this sort of structure. So this here, this is the bond that I was talking about down here. And this bond, so this is called a beta 1,4 glycosidic bond. Now it's probably a bit too much over your head right now. If you need more information as to how this bond is formed, look at page 58. 58 of your textbook will help you with this. And basically, because of this, you can have this provides it because these molecules aren't able to rotate around each other. Because of this, they have to invert every other one every 180 degrees. So what I'm trying to say is they need to every other beta glucose molecule needs to be inverted 180 degrees. Because of that, it provides a lot of structural support. It helps to provide support and rigidity, yes, for the plant cell wall. So basically this is just one straight chain of cellulose within a multi chain. So this is one chain, is what you see here. This is several hundred chains formed into a microfiber. And then several hundred of these microfibers form a macrofiber. And yeah, that's basically what you need to know for this. And because of this, cellulose has a very high tensile strength, and these macrofibers, myofibers, are able to crisscross with one another. So they are able to form this sort of structure. So they're able to like crisscross with these change of microfibers, microfibers able to like crisscross with each other and form like a rigid structure for the plant cell wall and provides like helps maintain its turgidity or like helps maintain strength. So because of this, it's a very good building material for the cell wall of plant cells because of its very high tensile strength. That's the main feature. It has a very good tensile strength. There are other structural cell walls that we need. So bacteria, I'm not going to go into it fully now, so actually I'll go into it a little bit more. So bacteria, so structural polysaccharide. We have a cellulose in plants, as I just mentioned. We have other ones such as chitin. So chitin is found in insects, so it helps provide the exoskeleton of insects, and it also helps, it's not a major component, but it helps in the support for cell walls of fungi, fungal cell walls. And we also have peptidoglycan, for bacteria. So that's the end of this first video of module 2.2 biochemistry. In the next video I'm going to go into lipids. So hope you can join me for that video, and I'll see you in the next video. Bye.